Hello and welcome to Sci Cam, the online science magazine show. I'm Michael Conterio, and with me tonight in the Cambridge Science Centre is Guyton Lee.
Hi there.
I'm going to be interviewing him about the setting up of the Science Centre and all of the fun exhibits that we've got here. And also speaking tonight, we have Emma Pewsey. Hello, Emma.
Hello.
And Sonja Dunbar.
Hello.
But we're going to start here. So, Guyton, can you just explain what is this wonderful venue that we're standing in?
So you're here today in the Cambridge Science Centre. We've been open now for about 6 weeks, and this is Cambridge's first hands-on science centre. We're open all year round. Now, you think about the centre of Cambridge. There's great science, great technology, not just in the university, but everywhere. But there's not been a place really open all year round where people, and families, and tourists can come along and have a play. So that's what we try to do.
Can you tell us a little bit about what was involved in actually getting this whole place set up?
A fair amount of blood, sweat and tears. But there's a lot of people involved in the project. There's our two founders who helped us get the money and sponsorship into here. There's also the back team as well, who did a lot of the exhibit building, well, pretty much all of the exhibit building, and also setting up at the centre. And hopefully a little bit later you will have a little sweep of the area with the webcam and see how that all looks, because it's a really impeccable, beautiful space. We're not as large as we want to be yet, but it's a development thing, and we hope in years to come to get to something even bigger.
You really had to hit the ground running over these past 6 weeks, though, haven't you?
Oh, absolutely. Our first week was half term, and during half term it was insanely busy, and but great fun. And the families that came in came back again. You know, we've had great feedback from people.
Oh, well, that's always a good sign if you're getting the families back. It's all about really getting the kids excited, isn't it? If we're honest, that's amazing.
Well, it's true. However, just the other night, we did an event just for adults. So, you know, kids are important, but actually, why should we be stopped from having fun and enjoying the science in a hands-on way as well?
Yes, and we're going to be demonstrating one of these wonderful modular tables that you've got that can wheel around everywhere.
Absolutely, yes. I'm going to be working my arm muscles quite a lot in showing this to you, but yeah, absolutely.
Okay. If you've got any questions that you'd like to ask Guyton, you can stick them in the comments on YouTube, on Google+, or even if you're watching us through the Facebook page. Stick them on there, and we'll try and put them to Guyton later on. But for now, we're going to go over to Sonja Dunbar, who's got a beginner's guide to photosynthesis. Hello, Sonja.
Hello.
What have you got to tell us then?
Plants. Without them, most of the organisms we're more familiar with wouldn't actually exist. They sit at the bottom of the food chain, eating the sun, if you like, while the rest of us eat them or other creatures that depend on them. They are the world's producers. Oxygen is merely a byproduct to them, but actually they manage to change the atmosphere on Earth on a global scale, which allows so many of us to exist and respire. And in today's beginner's guide, we'll be opening the window on the process that allows them to be that foundation centre of life on solid ground, photosynthesis. If I just quickly share some diagrams with you, this will hopefully make life a little easier. So, some of you may be aware of the general equation for photosynthesis. That is, you've got carbon dioxide and water, and with the help of light energy, they get converted to sugar and oxygen. You might even have heard of the pigment chlorophyll, but we're going to look at this process in a little more detail. So, in plants, photosynthesis occurs in organelles called chloroplasts. So they're little structures inside the plant cell. And if you zoom even further inside them, they have little membrane-bound bubbles that we call thylakoids. There are two main parts to photosynthesis, actually. It's not just one quick reaction. And those are the light-dependent and light-independent parts. And the first part is the light-dependent reactions, which I'll be talking about now. And they occur in the membrane of those thylakoids that we just mentioned. So, when you expose a leaf to sunlight, the photons that is, the discrete little packets of light energy, reach the leaf and are absorbed by a large array of pigments. And these pigments are usually chlorophyll, and there can be others, which is why you get sometimes plants with different coloured leaves. And the chlorophylls make up what we call a light harvesting complex. So they form this sort of almost solar panel-like array where they capture the photons and then they bounce the energy between them until the reaction centre in the middle is able to accept the energy input. It's sort of like an energy funnel, really. And this little clever network lets plants be very efficient at harvesting the light they need, but it has to be spaced out perfectly in order for it to work. And the reaction centre contains a chlorophyll that is held in a particular environment that slightly alters which wavelength it absorbs best, which means it's perfect to accept the photons that have lost a bit of energy having been bounced around the LHC. And when that reaction centre chlorophyll absorbs the light energy, it causes it to lose an electron to an acceptor in another part of the reaction centre. And that event is referred to as the photochemical event. The reaction centres are actually bound to a part of photosystems. And plants have two photosystems, named 1 and 2. And they are made up of many subunits, which also have other accessory type proteins and cofactors bound. Now, photosystem 2 actually comes first in the process of photosynthesis, and included among its cofactors are several manganese atoms, forming what's called the oxygen evolving complex. So this little collection of 4 manganese ions is able to split water into oxygen, protons, and electrons. And thanks to this cluster, photosystem 2 is the only known enzyme capable of splitting water. And we have a hard time engineering a device that can do so with such little energy input, which is perhaps not surprising given that researchers haven't even quite fully worked out how the water splitting occurs in photosystem 2. But we do know the destinations of all the products. The electrons go on to replace those donated by our previously mentioned special chlorophyll in the reaction centre, which sort of recharges it so it's ready to donate electrons again. The oxygen is just a waste product. It's released from the plant via the stomata, which are openings in the underside of the leaf, which is the same place that the carbon dioxide the plant needs gets in. And the protons stay inside the thylakoid and help build up a gradient that we'll come back to in just a moment. So first, we actually want to follow the donated electron. And so when it receives the light energy input on the far, the bottom corner of the diagram, the photosystem basically, it gains energy from this. It becomes excited, we call it. And the electron is then lost from photosystem 2 and passed, following the red arrows, through a whole bunch of different other proteins in what's called an electron transfer chain. So I use the word down to describe it being passed down this chain because of how we draw it out. And you can see, hopefully on your screen, the Z scheme, which is how we refer to this chain. And it's based on the redox potential of the components involved. So redox potential is a measure of the tendency to accept an electron. In the chain, electrons can only be passed to a component with a higher tendency to accept an electron than the component donating the electron, which you can see on the sort of axis on the side there. And so it travels down from photosystem 2 through several of the proteins until it reaches photosystem 1 in its unexcited state. And all this transport of electrons causes a very important thing to happen. It moves more protons into the thylakoid. And that's where the protons from the water splitting are also stored. So this creates a very powerful gradient across the membrane, because if you remember I said this occurred in the membrane of the thylakoids. And another enzyme called ATP synthase lets the protons flow back across the membrane so that the gradient is evened out. And it uses that travel, the transport of the protons, to drive a motor, which drives the production of, well, probably the most important molecule in this process, which is called ATP. ATP, while it might not be in our original equation, is actually the energy currency of the cell. And then you have photosystem 1. So if you remember, our electron had reached photosystem 1 in its unexcited state. And just like photosystem 2, it gets excited by light. And that changes its redox potential, pushing it back up in our Z scheme. So now you see where the Z shape on its side is coming from. And then it can start donating the electron down a second electron transfer chain. And the end fate of the electron is to allow the cell to make a compound called NADPH from NADP. And NADPH is the other thing that the cell really needs for later on. It uses it as a donor of electrons in the dark reactions, or the light-independent reactions of photosynthesis. And so we can see that nicely summed up in the light-dependent reactions in this diagram here. So we've split water into oxygen, electrons, and protons down in the bottom corner, and we've stored the light energy that's been absorbed at different points in a chemical form as ATP and NADPH. And from there, we move on to the light-independent reactions, also known as the Calvin-Benson-Bassam cycle, after the three men that discovered how it works. So the first step of the light-independent reactions involves an enzyme called rubisco. Rubisco is actually the most abundant protein in the world, as far as researchers can tell. And this very important enzyme does the job of fixing carbon. Now what that means is it combines carbon dioxide from the atmosphere with its 1 carbon molecule with a 5 carbon molecule, which is called ribulose 1,5-bisphosphate, forming a 6 carbon molecule, which actually splits into 2 3-carbon molecules. And it cycles round to do this several times so that you can form a 3-carbon sugar. Once you have your 3-carbon sugar, you can basically bind it to another 3-carbon sugar and form 6-carbon sugars, which hopefully makes sense. But the 6-carbon sugars have common names that we're a bit more familiar with. One of the most common one is glucose. So these are the sugars that we're after from the reaction of photosynthesis. And to do that, to push around the cycle and not only combine the carbon dioxide to make the sugars, but also to regenerate the initial compound that we bind the carbon dioxide to, the cycle has to go through 9 ATP molecules and 6 of those NADPH molecules that we made earlier to create a single 3-carbon sugar, because the cycle has to go around several times and incorporate several molecules of carbon dioxide. So now we've got everything we wanted out of the reactions. We've used light and water in the light-dependent half of the reactions, releasing oxygen. And then we used the carbon dioxide to make the target sugar in the light-independent reactions. In all that time, we used the energy stored from light captured by the chlorophylls in the chemical form of ATP and NADPH. And that is a whistle-stop tour of what is actually going on inside all the green plant cells. So perhaps now you know the chemical complexity, you'll give your salad a little more respect. And back to you, Michael.
Thank you very much, Sonja. Thank you very much, Sonja. There was a lot more to that than I'm sure not just me, but many people would ever have imagined. But yes, if you've got any questions for Sonja, again, just drop them in on the YouTube comments or the Google Plus comments, and we'll try and get around those at the end of the show. But I'm back here in the Science Centre with Guyton with one of his demonstrations to show off.
Well, not just mine. It's well, there's a lot of Dave Ansell from the Naked Scientists in this particular demonstration, in this particular exhibit, in fact. Would you like to take it with me at all?
Yes, please.
Okay. Well, more importantly, we're going to ask you to do a little bit of muscle power and work on this. So, for those of you who might not be able to see, inside here we have a whole range of magnets that are on a sort of like a turntable, and that's what we're turning when we turn this handle. Above them are coils of wire. So basically, it's a big electric generator. Michael, would you like to give it a spin? So at the moment, Michael is not having too many problems because, well, he's not even using it, because nothing is connected. Electricity can't flow, so it's not doing very much. However, we're going to put some things on to make it a little bit more difficult. So Michael, if you can give it a turn, keep on going. I'm going to slowly, something wrong, put on, so again, so you seem to be slowing down there. 3 lights. I think we'll put the fans on as well. We've only got one fan at the moment, Michael. You're not really trying hard enough. And now we'll put on something that's really difficult. This is the fan over here, which requires even more electricity being generated to activate. So at the moment, Michael's working at the fair old sweat, and I could let him do this a while longer, depending on how I feel. But I'm going to slowly switch things off again. Michael, what happened there?
All the resistance just went out of it all at once.
And your hand just went like that. So this is a wonderful exhibit for many, many reasons. For myself, it's a very haptic thing. It's very hands-on, and you feel the difference. You can feel the forces in action. And that's one of the most important bits, I think, of hands-on science. And in particular, you just get to see people just being frustrated and intrigued, trying out lots of different combinations. There's a fair degree of experimentation and testing with this as well.
Yeah, because that's what you're aiming for here, not just to like tell people about science, but to get them to actually give it a try and actually come to understand it themselves.
Yeah, absolutely. I mean, we're not a schoolroom. We don't want to be a schoolroom. We want to be a place where science can be experienced rather than sort of via a teacher up at the front. So actually giving them a chance to get, for lack of a better phrase, hands-on with the stuff is really, really important.
And there's so much in this one experiment, because you've got the creation of electricity, where you've got all the coils that the magnets are going past to create electricity. But you're actually feeling the resistance that you get when you've got a circuit going that you actually have to overcome. You have to put the energy in to...
Yeah, there's lots of different levels. On a very basic one, yes, you're making electricity, but then you can go into electromagnetic induction. You can go into sort of electric braking type stuff. There's lots of different levels in which you can pitch this.
Oh, yeah. So we don't actually have time to go through all the experiments here, which is a shame. But we'll have a little run around with the webcam in a little bit,
Sounds great.
And you'll be able to explain what else.
Absolutely, yes, sounds great.
But now we've got Emma Pewsey, who is going to be talking to us about the strange case of the spontaneous hip fractures. Over to you.
Hi. So as it is the Cambridge Science Festival this week, and our guest director this year is Benedict Cumberbatch, I thought I'd give a bit of a Sherlock Holmes mystery. I'd bring a Sherlock Holmes mystery to Sci Cam. The case of the spontaneous hip fracture. A lady turns around after a visit from a GP to say goodbye, and as she does so, there's a crack and she falls to the ground. Another lady is sightseeing in Cambridge when suddenly her hip gives way underneath her as she goes up a small step. And these aren't the only victims. Every year in the UK alone, Around 4,000 people have their hips break for no discernible reason. And now hip fractures are seriously debilitating. Breaking your hip isn't like breaking an arm where you just put it in a cast and a couple of weeks later everything's fine. Repairing a broken hip requires major surgery, and the trauma associated with the hip fracture is so great that around 10% of patients will die as a result. So a serious case to solve, and luckily we've got an expert team of investigators trying to solve it. So researchers from the Cambridge Bone Group, the University of Cambridge Engineering Department, and the Department of Rheumatology at Charles University Prague have got together, and using Sherlock Holmes' signature methods of careful observation and logical deduction, they found some clues which mean that the misery of hip fractures might be prevented for thousands of people in the future. So let's start off by looking at the evidence. So we need to have a look at some bones. Now bones can be thought of as nature's scaffold pulls. I've got a very crude model of a bone here. It's just a tube with some sponge in, but it's kind of like a normal bone. So in the centre you have some spongy bone, which doesn't contribute too much towards the supporting part of your skeleton. And the outside is a narrow, thin layer, a few millimetres thick inside bones, of densely packed minerals. And this is known as the cortical bone. Now, unfortunately, as we get older, our cortical bone tends to get thinner and weaker. And in severe cases, if it gets particularly thin and weak, this is known as osteoporosis. Now, the amateur detectives amongst you might think you've solved the case of the spontaneous hip fracture already. Perhaps osteoporosis is the cause of these fractures. Unfortunately, you're not quite right. In fact, it turns out that around half of people who have a fractured hip don't suffer from osteoporosis. So there's another factor at play. And this is where the careful observations of our investigating team come into play. So they looked at the hips of some women who'd broken their hips using something called a CT scanner. This is a standard piece of equipment in many hospitals these days. But the problem with the standard CT scanners in hospitals is that the resolution of the images they produce isn't quite enough to be able to determine the small changes in thickness of the cortical bone. But this didn't stop our investigators. They invented a new way of processing the images collected by such scanners, which is so good that it meant that changes in the cortical bone thickness of just 30 microns could be detected. Now, 30 microns is tiny, about the size of a grain of dust. So equipped with their careful observations, what did they find? Well, first of all, they found that the cortical bone of women who'd had a fractured hip was generally thinner than in people who hadn't. But they found an even more interesting result when they looked at the femoral neck region, which is the region of the hip where hips most commonly fracture. When they looked there, they found small patches of what they call focal osteoporosis. So these are small areas, maybe up to the size of a thumbnail, of the cortical bone, which are up to 30% thinner than the surrounding area. And these provide perfect places for a crack to form. And again, I've got a small demonstration with some cardboard tubes. So here is my cortical bone again, or sorry, my bone again. If I push on the sides, you can see it doesn't bend. But here I've got a bone, where in the black region, I've made it a bit thinner. If I push on that, the bone collapses, and if it was natural bone, it would break. So where does this leave the case of the spontaneous hip fracture? Well, the case isn't quite closed just yet. Further studies will be needed to show if these focal osteoporosis patches actually do cause hip fractures. However, if they do, they lead to the tantalising possibility of a future where we might be able to treat hip fractures before they even occur. And subsequent studies have shown that by treating patients with the focal patches using standard osteoporosis drugs, it's possible to strengthen and thicken those patches. And remember, not everyone, or in fact most people with the focal osteoporosis patches don't have general osteoporosis. By targeting them with drugs, we might be able to prevent their hips from breaking. That means thousands of people every year won't have to go through the misery of hip fractures. Thousands of people get to have longer and better quality lives. Now I think that is a result that even Sherlock Holmes would be happy with. Back to you at the Science Centre.
Thank you very much, Emma. We are going to take a brief whistle-stop tour of the Science Centre through the medium of moving a webcam.
Yes, let's hope that technical doesn't hit any technical glitches, but we'll give it a try.
Yeah, make sure we've actually got the right webcam on. There we go. Yeah. So, Guyton, can you just point out a few things in here?
Yeah, absolutely. So, if we get the camera steady, let's have a look on this sort of far wall over here. So we've got a variety of things. I will sort of point with my finger just over this way. We've got our wonderful sort of pinhole cameras over here, sort of showing about how the eye has evolved. We've got other things about over this way, all about sort of electricity and magnetism as well. One of the things that we're very keen on with our exhibits is that they all tie and link together in some way. And you may not be able to see them, but all along the wall, we've got a whole range of panels as well that talks about local science. This is Cambridge Science Centre. We want to celebrate all the great science that's happening here in town now and in the past. So we're very much keen on sort of trying to bring up those local stories from the university, from the research institutes, and all those sort of companies as well, because there are a great number of those doing amazing work all around us. And oh yes, over here. Well, we've got our sound demos just over here, building bridges. We've got light. And I mean, my personal favourite just over here is our engineering wall. If that's focused in about the right place, yes, I just move it slightly to your right, Michael. Up. Yes, so over here, this is our engineering wall, and on here, there's a whole lot of gears, and this is what we really love. It's open-ended play, it's experimentation. They build, they choose what they want to get out of it. We can come along, help a bit more, but they can build whatever machine they want, they can hook it up in whatever ways they want, and that's a really powerful thing.
If people wanted to come here, when are you open?
So, during term times, we are open on Saturday and Sunday, 10 till 5. During the week, Mondays we're closed, and then Tuesday to Fridays in the afternoons from 1 till 5. During the holidays, half terms, etc., we're open Monday to Sunday, 10 till 5.
Well, thank you very much, Guyton.
No,
we can just come back round here and invite Sonja back as well into the hangout. Have you guys got any questions that you'd like to put to Guyton? Hello? Emma and Sonja, are you there?
I'm here.
Hello.
Hello. Have you got any questions for Guyton?
So I was going to ask. You said that like the experiments seem to be like linked to each other. Is there like an overarching theme of the experiments at the centre at the moment?
So this is our opening exhibition, and for the opening exhibition, what we really wanted to do is sort of present a general showcase of science, but tying into general Cambridge research as well. We are going to be looking to do more themed exhibitions when we move forward in the next year, and hoping to have a new exhibition over the summer.
Wow.
Emma, I'd actually like to ask you a question. You were talking about the drugs that people were taking to treat osteoporosis and how they could be useful dealing with the hip fractures in people who don't have general osteoporosis. How do these drugs work in general?
I think they mostly act to sort of remineralize your bone. So osteoporosis arises because there's an imbalance in the processes that occur in the cortical bone. So throughout your lives, you get new bits of bone being laid down and old bone being destroyed. That's old and worn out bone. And in osteoporosis, these two processes get imbalanced, and so the bone is destroyed faster than it's created. And so the drugs act to sort of add in some extra minerals and strengthen it like that, I think. Not too sure.
Guyton, you had a question to ask Sonja.
Yeah, I did. So, Sonja, I mean, I always find it fascinating that research scientists are trying to look at ways of genetically modifying plants to try and get more yield out of them. Photosynthesis has evolved over a very, very long time. How much wiggle room is there to actually improve on that process, or is it pretty much as good as it can be?
So there's a little extra detail that I didn't actually get a chance to mention because we'd already covered a lot of ground. And there are actually slightly varying types of photosynthesis, which we call C4, C3, and CAM. And they incorporate carbon in different ways. And some of them are a little bit more, I guess, careful with the resources they allocate. So actually, if you're looking at trying to improve yield, if you're making crops for, say, an area where you know you're going to be like heavily fertilizing them, so they're going to have all the nutrients, all the water they could want, then actually you'd want something like the C4 strategy. Whereas if you were, say, trying to engineer a crop that would give a very good yield in, say, like the harsher regions of maybe Africa and places like that, then you might try and engineer a slightly different strategy so they'd be more resilient. So there's a lot of potential actually, and there's some fantastic work going on in the Cambridge Plant Sciences department at the moment, trying to look at improving crops.
Thank you very much, Sonja. And that brings us to the end of our show. I'd like to thank Gayton for letting me in and showing me around his wonderful science centre.
Not a problem.
And I'd like to thank Emma and Sonja for their interesting talks. Thank you very much, guys.
Bye.
And we'll be back with another episode of Sci Cam sometime after Easter. Follow us on Facebook, follow us on Google+, and we'll let you know. Thank you very much, and good night.